Good afternoon, everyone. My name is Sophia Jean V. Coyoca, and my partner's name, Luisa Marie C. Sabello. Our thesis advisor is Nanette D. Santos. Our topic is Water Quality Assessment of Taal Lake, an integrated approach to microbial and chemical contamination. For introduction, our research is titled Water Quality Assessment of Taal Lake, an integrated approach to microbial chemical contamination. This study explores the environmental health of Taal Lake, a significant recreational lake in the Philippines. It's an essential natural resource for local livelihoods, tourism, and leisure. Unfortunately, contamination, particularly from microbial sources, poses serious health risks for both visitors and the local population. These risks arise from untreated sewage, agriculture runoff, and animal waste, all of which introduce bacteria, viruses, and protozoa into the water. Our study aims to provide a comprehensive analysis of the levels and potential health impacts of microbial and chemical contamination in Taal Lake. Using both microbiological and physicochemical parameters, ultimately, our goal is to provide critical insights that can guide water quality management practices and ensure the safety of Taal Lake for recreational use. For the significance of the study, first, determination of the presence microbial contamination and chemical characteristics such as pH, turbidity, temperature, BOD, BO, TTS, nitrates, phosphorus, total coliform, and E. coli. Second, raises public awareness about Taal Lake's environmental health. Third, aims to provide an integrated assessment of the environmental health of Taal Lake. Lastly, recognizing its significance to the lake water, aquatic life in the lake, and the individuals that live around the lake. For scope and delimitation, the study will focus on assessing water quality around Taal Lake, specifically targeting areas near human activities like recreation and fishing. It will examine microbial contaminants such as E. coli and fecal coliforms alongside key water qualities factors like pH, dissolved oxygen, and heavy metals. Seasonal changes will be considered to capture variation in contamination across dry and wet periods. Data will be gathered through the water sampling and lab analysis with additional context provided by existing data on pollution sources. The limitation includes focusing on select contaminants and excluding groundwater sources, longitudinal studies, and in-depth health analysis. Budget limitations may also affect sample size and lab testing scope. The study aims to deliver valuable insights on microbial contamination risk and water quality while acknowledging its specific focus and constraints. For the objectives, establish a thorough assessment of the lake's water quality by combining physicochemical and microbiological contamination. Provide reliable information to support regulatory policies and assessments on water management. Measure the levels of coliform bacteria with a particular focus on E. coli in the lake water. For methodology, our study focuses on water quality in Taal Lake with sampling conducted in a designated site near Batangas City. We, the researchers, selected this area for its sustainability for monitoring. Two types of tests were performed, one for physical and chemical water quality and another for microbial contamination. The samples were collected and analyzed between September to October 2024. The collected samples between 9 to 9:25 a.m. were placed in three different bottles, which are 120 ml for microbial contamination and 1 liter, and 3 liters for physical chemical tests, and we put it inside the ice box cooler with a lot of ice. Water samples were analyzed using the APHA AWWA method, following guidelines from standard methods for the examination of water and wastewater. Findings were compared with the standards from General Effluent Standards of 2016 and DAO number 2016-08 Water Quality Guidelines. Test includes physical parameters like temperature, pH, dissolved oxygen, and total dissolved solids. Also, biological oxygen demand, as well as chemical parameters such as phosphate and nitrate. Next, I'm going to discuss the standard values set by DNR EMB. In the table 1 shows of all the physical and chemical parameters, standard parameters set by DNR EMB. For the first one, pH, it ranges 6.5 to 8.5. Temperature ranges to 24 to 27 degrees Celsius. Turbidity, at least 5 NTU. Total dissolved solids, not more than 30% increase. Dissolved oxygen, 5 minimum. 5 is the minimum. For the biological oxygen demand, 5 is the minimum and 10 is the maximum. For nitrogen, 7 milligrams per liters. Phosphorus, 0.5 milligrams per liters. Next, for the standard value of the presence of total coliforms and E. coli in recreational water standards based on WHO, and the specific guidelines or regulations set by local or national environmental authorities, here are some general guidelines. For recreational water, use guideline often set a limit for the presence of fecal indicator bacteria like E. coli based on who the standard values in recreational category 2, class A, B, and SB recommends at least 5,000 mpn per 100 ml of total coliform and at least 200 mpn per 100 ml of E. coli in the fresh water bodies. It is interpreted non-polluted. For the results and discussion, the table shows the results of the water analysis parameters of Taal Lake at Club Balay Isabel, Talisay, Batangas is Site 1 and Taal Central Fish Port, Laurel, Batangas is the Site 2. For the pH scale shows how acidic or alkaline the lake water. It is determined by the overall pH level, whether the aquatic life is safe or not. With the low pH level in rivers and lakes, can kill a lot of sea species, and since some of them are sensitive to the pH changes and some may not be able to survive. Example, with a pH level of 4 or below. With the set of the results of an average pH value of 7.68, it is safe and meets the DNR EMB standard values. For the total dissolved solids is 1,025 milligrams per liters. It is quite near to the standard set values. TDS provides an overall impression of the purity of the water by measuring all dissolved elements in it. Therefore, it is essential for healthy ecosystem. For temperature, the reported average temperature is 20.45 degrees Celsius. It is below the set value limit because the ambient water temperature ranges in lake is 25 degrees Celsius to 29 degrees Celsius at the surface, depending on the season. This temperature range is impacted by Taal lakes. Tropical location and environment. Natural changes within this range are generally acceptable for ecological health unless considerable human activities include further warming as the DNR EMB normally permits. For the turbidity, with the result of 2 NTU, which it is below the standard limit value set by DNR EMB, the average value has low level of turbidity which the lake water is clear. And for the phosphate, the average phosphorus concentration is 1.005 milligrams per liters. It is higher than the standard values by DNR EMB, which is 0.5 milligrams per liters. Algae blooms can be supported by high phosphate levels and enough dissolved oxygen. Eutrophication is one of the main causes of this. The water quality degrades, which affects recreational activities like fishing, swimming, and etc. It can also contribute to the growth of harmful algae, which can be toxins and can be harmful to humans and aquatic life. The average nitrate concentration is 0.364 milligrams per liters, which is very low than the DNR EMB limit
set of 7 milligrams per liter. Reasonably low nitrate level indicate a little contamination from the sources. It can be sign that the environment is balanced, not unduly impacted by human activity, and is home to a wide variety of species. For BOD, that is not detected, it usually indicates that there is very little organic matter present in the amount it is below the limit of what the testing apparatus or equipment can detect. For the dissolved oxygen, is content is 8.05 milligrams per liter. It is above within the acceptable range. It means that the water quality is good for the majority of aquatic ecosystem. According to the overall findings, the Taal Lake water can still be categorized as recreational water class category B. It is a good place for fundamental contact activities, including swimming, fishing, skin bathing, and etc.
For microbial contamination test, the table presents total coliform counts from water samples taken at Taal Centerfish Port and Club Balay Isabel Resort using the APHA and AWWA method. These measurements reveal the presence of coliform bacteria, which can indicate contamination from fecal matter and suggest potential health risk. At Club Balay Isabel, the coliform count in was recorded at 1,600 mpn per 100 ml. This level meets standards for safe drinking and recreational use, indicating a low risk of waterborne disease. At Taal Centerfish Port, the total coliform count was 350 mpn per 100 ml, which also falls below the DNR EMB standards, showing no immediate contamination concerns. Both sites are classified as non-polluted based on these results. The data implies that while current levels meet safety standards, continuous monitoring is necessary to maintain water quality. Further investigations and actions may be required to ensure long-term safety, especially since coliform levels can fluctuate. The BOD was also measured to assess organic pollution. Low BOD levels indicate that organic matter in the water is minimal or below the detection limit. This means the oxygen demand by microorganism breaking down organic substance is low, further supporting the conclusion that the water has minimal contamination. Regular testing and adherence to local water quality standards are essential for both locations to guide any needed water management interventions. For conclusion, a thorough health risk assessment for Taal Lake is essential to ensure public safety and sustainable management for this vital resource. Regular microbiological testing combined with monitoring of physicochemical factors like pH, temperature, and dissolved oxygen provides a clear understanding of contamination sources and potential health risks. Findings indicate that while some water quality parameters are within safe limits, volcanic activity has led to high microbial contamination levels, making the lake temporarily unsafe for recreational use. Continued collaboration among stakeholders is crucial for effective action planning, public education, and regulatory measures. By grounding these efforts in accurate water quality data, we can protect public health, support safe recreational and aquaculture activities, and promote long-term sustainability of the lake. That's it for our presentation. Thank you for listening.